Hi, so I'm Manu De Rycker. I'm the head of biology in the drug discovery unit at the University of Dundee, and I look after our kinetoplastid drug discovery program. Yeah, so Chagas disease is called a silent disease, in my mind really for two reasons. I mean, there is the sort of biological reason, which is that when a person is infected with the parasite that causes Chagas disease, called Trypanosoma cruzi, people are unaware of that very often. And so people can live for 10, 20, 30 years with this parasite inside their body without knowing it. And then eventually symptoms might occur, which is sort of the result of damage that happens over the years. And so it's a silent disease because you can live without knowing that you have the infection. But it's also a silent disease, in my opinion, because very few people know about it. Patients don't know about it necessarily. Funders don't know about it necessarily. So yeah, two reasons why it's silent. I think even though we don't know very much, many people don't know about it, about 10 million people are thought to be infected by this disease. So, that's almost twice the population of Scotland, which is staggering, really. Yeah, so the biggest issue currently with the existing treatments for Chagas disease are the side effects that come with these treatments. So patients often stop their treatment early because of these side effects. So sometimes doctors don't even prescribe the medicines because of the side effects. So that clearly is a very important issue. And it's the main reason we need new and better treatments. So progress has been made. So several groups, including ourselves, are progressing compounds towards new treatments. But it is very challenging. There's only a handful of groups working in this space. And so I think there's a lot more work to be done, especially when you think about attrition. So attrition is the fact that compounds, new treatments, can fail at any point on the way to patients. And so for that reason, we need to make sure that we keep replenishing what we call the pipeline with new potential treatments to make sure that eventually we get to something that will deliver real benefit for our patients. Making treatments for Chagas disease has some really particular challenges. And one of them is the fact that we have very few parasites in the body of people that are infected. And it's that very small number of parasites over many, many years that causes the damage that leads to symptomatic disease. And so the big challenge is that we all kind of agree that we need to kill every single parasite in a patient to prevent disease from occurring, to prevent symptoms from occurring. Now, killing every single organism is difficult, and we found that it's particularly difficult with Trypanosoma cruzi, for which there tend to be some parasites that can survive treatments. And so once we find that treatment that can kill every single parasite, then we really get excited about that. A second challenge is that even though there aren't that many parasites in the body, they can go anywhere in the body. They can infect any tissue in the body. And so from a drug discovery point of view, to make a new treatment that goes to all parts, all tissues in the body, and is very safe, is a real challenge. So altogether, having to kill every parasite and reaching all the different parasites makes for a difficult puzzle. Yes, so we have been working 20 years in kinetoplastid drug discovery right now, and we have learned a lot along the way. We're probably a little bit naive initially with some simple approaches, relatively simple approaches to what we were doing. But over the years, things have become a lot more sophisticated. We have learned a lot from failure, as you do. When experiments don't work, you try to understand why, you try to improve what you're doing. I think one of the best examples of that are the clinical trials carried out in the 2010s with compounds like posaconazole. So these were compounds that moved forward to patients in phase 2 clinical trials, and they failed badly in the clinical trials. And of course, that's kind of a wake-up call, right? So drug discovery produces a compound, and then ultimately it doesn't work. And so even though that was a failure, we and all other groups involved in Chagas disease drug discovery have learned a lot from that. And we have started developing new laboratory models that can improve our predictions and avoid these kind of failures in the future. So a key example of that is different laboratory assays that can assess whether a compound can kill every single parasite rather than leaving a small percentage alive. And so we call them our washout assays, and they have made a big difference in our ability to predict where we're going. So we have now a complicated set of multiple assays that all contribute to predicting the outcome, and I think that is probably one of the biggest changes compared to where we started. So yeah, when developing a Chagas treatment, we start with compounds, small chemical molecules, and we check whether they can kill the parasite. And so initially, if you see they kill the parasite, that's great, but it's not sufficient, because what we want is a compound that eradicates every single parasite. So that's, when we generate data for a compound series that it can actually kill every single parasite, that's when we get really excited and really get going on pushing that drug discovery project forward. But you do not only need, of course, to be able to kill every parasite, you also need to be able to make it, say, in a pill. So we need to show that this compound can be used orally as a simple pill. And then finally, and really important, is safety. So we want to make a treatment that is much safer than the current treatments. We want to make a treatment that you can give to people that have absolutely no symptoms and doesn't make them feel worse. So yeah, these are some key points for new treatments. Yeah, so drug discovery in general is a very challenging process. So, frequently compounds fail. You might have been working on them for a year, for 2 years, and then some piece of data comes around and says, okay, well, you can't progress this. So that is difficult, but you need to make these difficult decisions. The resources we have in this space are very limited, right? And so it's absolutely critical that we focus our resource, our people time, everything on progressing only the most interesting compounds. So these are tough decisions that we have to make, and we have to make them on a daily, weekly basis, where we get together with the team, with all our experts, and we decide whether it's still worth progressing, whether we still think we have a good chance of delivering a new treatment, or whether the time has come to say, okay, we have to stop this now because we can better spend our time on another option for Chagas disease. Yeah, so there are many pivotal moments in drug discovery programs. A lot of these are kind of negative results. They're not the results you want to see, they're disappointment. But of course, they put us on track for the next step to move forward. But of course, there are positive breakthrough moments as well. And I think we've talked about how we want to achieve full cure. We want to be able to kill every parasite. So when we run these experiments to check that, these are like 2-month-long experiments. When at the end of that experiment you see, oh, this compound has worked, it has, there's not been relapse, it has killed every single parasite. These are exciting moments for the project. But there are other kinds of breakthroughs. An example I can think of now is when we went to a conference in Bolivia and we saw a presentation from another university showing a really interesting compound series. Was very exciting what that could achieve in Chagas disease, and so we started talking with each other, and that has led to a long-standing collaboration now with the University of Washington, who are presenting that data, as well as DNDi, the Drugs for Neglected Diseases Initiative. And together right now, we are pushing forward to try to get to a new treatment for Chagas disease. Collaboration is absolutely essential in this space, right? So we have limited resources and everybody has their own expertise. Now, drug discovery is a very long and complex process, and so you need to have understanding of the basic biology of your parasites. You need to understand how to do early drug discovery, late stage drug discovery, and ultimately clinical trials and registering the compound getting to patients. So a long, complicated and expensive process, and nobody can really do this on their own. So in our work in Chagas disease, and in general in our neglected tropical disease work, we always work with collaborators. They bring all their expertise to the table. It makes it very exciting because you have all the people that have the knowledge required to successfully deliver the program. So collaboration is essential and it's just also very enjoyable. So I think, to improve the lives of people with Chagas disease, what we need in the first place is a new treatment, is a better treatment that is safe, which is the issue with the current treatments. Once we have a safe treatment, well, the diagnostics are in place to find people that have the disease, but there has been little incentive to go and find all the patients because we don't have good treatments. So once we have good treatments, there will be an incentive to start looking for people that are infected and then treat them. And so I think new treatment will lead to a change in public health systems to identify patients rapidly, then treat them and make a real difference. So yeah, looking ahead 10 years into the future, what I really hope is that the work we've been doing in the last 10-15 years on developing new treatments will bear its fruits. And what I mean is that I hope our compounds will have made it to human clinical trials. In particular, phase 2 clinical trials where you check whether the compound shows efficacy in patients. So even though 10 years sounds a long time, these processes take a lot of time getting through all these stages. So if we get there in 10 years, I'll be very excited. More widely, for everything we do in the drug discovery unit, so we have a range of clinical candidates. So these are compounds that have made it to clinical trials that are in people for testing. But what we really want to see next is that these compounds become registered medicines, so that they are actually there available, in hospitals, in pharmacies, to treat the general population that have these infectious diseases. So yeah, looking forward to what's going to happen in the coming 10 years.